Dear friends, today I'll explain you about monoclonal antibodies which can be used as treatment for covid-19. In the composition of blood, plasma is there which is having formed elements like platelets, then white blood cells or leukocytes, RBCs or erythrocytes. Now again in this diagram it's visible that this upper portion is having plasma which contains water, ions, proteins, nutrients, wastes and some gases. In between platelets and then red blood cells are there. WBC is mainly responsible to provide defense and it is involved in immune response also. Majorly these four cells of WBC, macrophages, T cells, these are also known as T helper cells, B plasma cells and B memory cells. It is more clear from this figure that whenever any pathogen, that is virus, bacteria, fungi, whenever any pathogen enters in our blood, their macrophages, these are big sized cells of WBC, they engulf the pathogen and then they send this information to T cells. Means T cells get activated. T cells help in recognition of antigen. So here antigen is the foreign matter, that is pathogen, which is recognized by T helper cells. T helper cells, they send this information to B cells. B cells in response, they turn into plasma cells and they form antibodies. One part of the B cell turns into B memory cells which memorize the form of antigen. So whenever same antigen attacks, then there will be no need of recognition by T helper and then sending information to B cells and then to plasma cells, then to antibody. Once antigens are already being, body is already being infected, in the second attack, B memory cells are there and they produce their immune response. So here antibodies bind to antigen. This is the normal process how immune system, how immune response occurs in our body as defense mechanism. Now, what is difference between antigen and antibody? Antigen is a substance which can induce the immune system to produce antibodies against it, and it is a foreign matter. It can be pathogen, virus, bacteria, and even during organ transplantation, foreign organ is also considered as an antigen for our defense system. Antibody is a protective protein which is produced by B cells of the immune system in order to attack antigens. Mostly antigens, they are made up of combinations of polysaccharides and antibodies are made up of proteins. These can be cells and antibody cannot be cells. So both of them act as key and lock. So if we have to check about types of antigens, so mainly there are two types of antigens, self antigen and non-self antigens. Self antigens, they occur whenever any autoimmune disease occurs. Whereas foreign antigens, these are non-self antigens like pathogens. In case of antibodies, there are 5 types of antibodies present in our body, that is Ig, immunoglobulin A, D, E, G and M. Now coming to the interaction or effect, how this antibodies, they are having effect on the SARS-CoV-2. Mostly what happens, this is outer membrane of human cell which is having receptors. So these are receptor cells. So in our body naturally we are having polyclonal antibodies, but SARS-CoV-2, it is so strong that it fails the human defense system, that is polyclonal antibodies. That's why we have to make monoclonal antibodies in artificial lab conditions specific against this specific antigen. So in this figure it is being shown that monoclonal antibodies, they'll bind to the outer protein covering of SARS-CoV-2 and preventing it to bind with the receptors of human cell. Again, it will be more clear to you from this figure. Now, in the first figure, when we don't have any monoclonal antibody, SARS-CoV-2 spike protein, it identifies ACE2 receptor and it binds with it and attacks the human cell. On the other hand, when the person or patient is injected with monoclonal antibody, which is specific against this receptors of the spike protein, then they prevent it from binding with the ACE2 receptor of the antibody, receptor of the human cell. How can we make monoclonal antibodies against a specific antigen, against a specific pathogen? Like these days scientists, they made monoclonal antibodies against COVID-19 virus. In this, they first take we have to take experimental animal system. Now, epitopes are actually antigens like coronavirus. It is injected, small amount of it is injected in the like in rabbit in the animal system. Then its spleen cells are taken because B cells are there which are required to form antibodies. Now we need large number of B cells. For this we have to fuse them with myeloma cells which can make them multiply rapidly, and this process is known as hybridoma, and for this we need an enzyme polyethylene glycol, and then from combination of cells, hybrid cells which are having combination of B cells and myeloma cells, they are taken, selected and used to prepare monoclonal antibody. It's not a cheap process, it's really difficult to make, and it's expensive also, but technical or skill is required for its formation. During this COVID-19 time, in US, actually FDA, they give approval of 2 monoclonal antibodies to use for the treatment of COVID-19. Bamlanivimab and itesevimab both are used for the treatment, and within 2 to 3 days these are used for mild, severe and for moderate infections, and within 2 to 3 days positive results they were found. Now, finally, what is the difference between antibody therapy and vaccine? Both are actually used for the, one is used for the treatment, another one is the preventive. Now, what is it? First of all, though, what is this monoclonal antibody therapy? Here we are making monoclonal antibody artificially against a specific antigen, that is actually COVID-19. This protein binds to the virus and stops it from infecting you. On the other hand, vaccine like Covaxin, Covishield, Pfizer, etc., it's in this a piece of virus is there which is injected in our immune system, and immune system can recognize that. Then what is it for? This antibody therapy is used for the treatment those who are already infected with COVID-19. But vaccine, it trains the immune system of the human system to fight against COVID-19. And how fast does it work? Antibody therapy, as soon as it is given to the patient, it will work, while vaccine within 1 to 2 weeks after both doses are given. And how long does the protection last? In case of antibody, weeks to months, whereas in vaccine, years to lifetime. So still research is going on in this field, and people are they are making monoclonal antibodies, and these are already being used in the treatment of organ transplantation. Then it is also used in the treatment of cancer, and now monoclonal antibodies are made against COVID-19 pathogen, against this specific antigen, to control its multiplication and number. And both, if antibody therapy and vaccine will be used, then it will give positive and good results. Thank you.